The human brainstem serves as the essential gateway for consciousness and the primary regulator for the modulation of sensory information. Early neurophysiology characterized this region as a diffuse and nonspecific network, a simple web of neurons known as the reticular formation. Contemporary research has dismantled that model, revealing the reticular activating system, or RAS, as a discrete architecture of nuclei with specific cellular morphologies and neurotransmitter types. This system acts as the biological regulator for the cerebral cortex, ensuring cognitive centers are sufficiently stimulated to maintain awareness, while managing the transitions between waking and sleep. Confined to a column just 2 inches long, the RAS coordinates ascending and descending pathways that influence nearly every aspect of central nervous system activity, from motor coordination to the electrical signatures of thought. It is not a passive receiver. It actively filters the sensory barrage of all 5 senses, allowing the brain to focus on vital information while tuning out redundant environmental noise. To understand the conscious state, we must map this specific physical circuit rather than treating arousal as a vague psychological phenomenon. The core of the reticular formation is organized into 3 distinct longitudinal columns. At the center lies the median column, primarily composed of the raphe nuclei, serving as a hub for serotonin synthesis, regulating mood and circadian cycles. Directly adjacent is the medial column, containing larger gigantocellular nuclei that facilitate motor coordination and modulate somatic muscle tone. The outer lateral column consists of parvocellular nuclei, which integrate sensory feedback to regulate autonomic functions like exhalation. These columns physically bridge sensory inputs from the spinal cord to the higher processing centers within the thalamus and hypothalamus. This spatial organization allows the brain to separate basic autonomic execution from the higher-level regulation of consciousness. While the columns provide the structure, the ascending reticular activating system is driven by a series of specific chemical engines. Arousal relies on 4 major groupings of nuclei, each releasing specific neuropeptides to create distinct flavors of awareness. The locus ceruleus releases norepinephrine across the cortex to enhance focus and wakefulness. The tuber mammillary nucleus serves as the primary source of histamine projections, vital for maintaining the alert state. And the pedunculopontine tegmentum releases acetylcholine to drive a process called EEG desynchronization. These high-frequency waking rhythms were first physically measured and documented during the early era of neurophysiology. EEG desynchronization occurs when cholinergic neurons switch the brain from slow, synchronized sleep rhythms to the high-frequency, low-amplitude patterns associated with alertness. Dopaminergic nuclei, such as the ventral tegmental area, then provide the reward-driven arousal necessary to transform simple wakefulness into motivated behavior. Consciousness is a product of this precise, tunable balance of neurochemical inputs. To reach the higher processing centers of the cortex, these chemical signals must travel through an intricate upward neural routing system. The thalamic pathway is the primary route for cholinergic neurons. It relays rapid signals from the midbrain into the thalamic relay nuclei, keeping the brain on by suppressing slow cortical waves. The second route is the hypothalamic or extrathalamic pathway, bypassing the thalamus entirely. By utilizing monoamines like serotonin and histamine, it provides a diffuse, long-lasting modulatory influence to support sustained attention. This dual routing system ensures the cortex is both awakened instantly and sustainably powered for behavioral engagement. The stability of the RAS network is governed by orexin in the lateral hypothalamus, projecting directly to the 4 chemical nodes. Orexin provides the essential excitatory drive to stabilize the system, preventing the brain from slipping into sleep. This drive is triggered by the environment. Specifically, 480 nanometer blue light is captured by non-visual photoreceptors in the retina, called iPRGCs. This light exposure activates the orexin neurons, which then stimulate the brainstem to boost alertness and improve mood. When this excitatory drive is removed, the entire arousal architecture begins to collapse. Type 1 narcolepsy is a severe structural deficiency resulting from the loss of these specific neurons in the lateral hypothalamus. A 90% loss of orexin neurons breaks the stabilization of the system. Even with rest, the brain cannot mechanically sustain wakefulness for more than an hour or two. Without an intact orexin system, environmental light signals cannot sustain the awake state, reducing arousal to fleeting, unstable bouts of wakefulness. Beneath the broad chemical pathways lie the intricate, microscopic electrical signatures of the individual brainstem cells themselves, firing in perfect sequence. Stable consciousness relies on gamma band oscillations running between 30 and 90 Hz. High-threshold calcium channels and GABAergic gap junctions electrically couple these neurons. This creates massive synchronized network coherence that allows the RAS to stream information to the cortex. To prevent this massive electrical activity from overloading higher cognitive centers, the brain employs an automatic filter. The thalamic reticular nucleus, or TRN, acts as the guardian of the gateway between the thalamus and cortex. It uses lateral inhibition to gate data. Attending to sound suppresses neuronal activity in irrelevant sectors, like vision. Failures in this gating mechanism, often evaluated using the P50 EEG wave, are biological markers for the distractibility and hypersensitivity seen in schizophrenia and ADHD. The RAS does not just turn the brain on, its subcircuits actively carve out our perceived reality by deleting irrelevant noise. This complex architecture requires absolute structural integrity for signal conduction. Myalgic encephalomyelitis, or chronic fatigue syndrome, reveals what happens when this internal connectivity fails. Recent fMRI data shows that in ME/CFS patients, the connectivity between the medulla and midbrain nuclei is significantly diminished. This localized deficit causes the entire thalamocortical coherence to collapse, resulting in unrefreshing fatigue and autonomic dysfunction. By mapping these discrete cellular structures and chemical gates, we understand human consciousness not as magic, but as an exquisitely engineered biological circuit.